sets with you today. I'll be talking about a novel technology which we have developed, which we call Avatar. It's essentially a bioreactor that allows for the precise modeling and mimicry of in vivo tumor microenvironments. Now, it's well known that tissues and tumors are intrinsically hypoxic, ranging from as low as 1% to 10% oxygen. Yet, majority of in vitro cell culture models rarely incorporate hypoxic culturing conditions into their workflow. This consequently results in gene and protein expression profiles that are not physiologically relevant, and this becomes problematic when performing gene expression analysis. Now, years of research by Greg Semenza at Hopkins, as well as others, have shown that low oxygen levels increases HIF-1 alpha expression, a master transcriptional regulator involved in vascularization, angiogenesis, tumor metastasis, and cell survival. In short, culturing any cell type at low oxygen levels promote the activation of multiple pathways, including VEGF, P53, and ERK signaling pathways. In addition to hypoxia, and perhaps less appreciated, is the impact of interstitial fluid pressure on gene expression. Seminal work by Rakesh Jain's group at Harvard Mass Gen has shown that interstitial fluid pressures are elevated on a cohort of solid tumors. This increase in fluid pressure results in activation and regulation of various receptor tyrosine kinases, engagement of integrins, and changes in glucose metabolism, as well as many other critical factors. So we decided to incorporate these important physiological features by developing a benchtop sized bioreactor that's stackable, as depicted on the right, allowing researchers for the first time to culture cells at various oxygen and pressure levels that reflect diverse tumor microenvironments, resulting in what we believe to be a physiologically relevant gene expression profiles. We then applied our technology to capture and propagate circulating tumor cells from peripheral blood samples of metastatic castration-resistant prostate cancer patients. After failing to grow enriched CTCs from other microfluidic enrichment systems where viability of enriched cells were quite low, we decided to use a well-established old-school technique, Ficol-based centrifugation, to collect the PBMCs, or the nucleated layer, containing your CTCs as well as your immune cells, and we plated these cells directly onto a growth factor-infused collagen-based hydrogel. Now, the rationale being that cells with epithelial-like phenotypes, aka carcinoma cells, would bind tightly to the underlying collagen, whereas the majority of the immune cells would not adhere to the substrate. 2 hours after plating, we perform a simple media exchange to remove the non-adherent cells, leaving you with a sort of enriched adherent cell population. So when we take a closer look at these prostate CTC clusters or colonies under the microscope, it's just a bright field image, we see that these colonies resemble mini tumors with tightly adhered and well spread morphology. The number of CTC colonies from 10 mils of peripheral blood varies from 0 to about 24 colonies, comprised anywhere from just a few cells to hundreds of cells. Now, what is interesting about these colonies is that from the same patient sample, we have morphologically distinct colonies. And this is also variable between patient samples. The detection of these colonies in prostate cancers was about 25%, while in non-small cell lung cancer is higher, ranging about 40%. Next, we decided to take a closer look at these colonies doing immunofluorescence imaging. And what we see is that they're often composed of heterogeneous cell types. These include immune cells. The immune cells here are depicted in green and indicated with arrowheads. Macrophages, dendritic cells, and T cells, just based on staining and morphology, are shown readily in these growing colonies. Next, we performed qPCR on individual CTC colonies that were collected by laser capture microdissection, or just simple micropipetting. We saw increased gene expression in markers associated with prostate cancer, and saw variability in terms of their expression across CTC colonies and across patient samples. Interestingly, a subset of these patients showed increased expression of PDL1 and CTLA4, along with stem cell factors OCT4 and SOX2, which were upregulated when compared to canonical prostate cancer cell lines, LNCAP and PC3s. Next, these prostate colonies were collected and profiled transcriptomically. And what we saw was an increased gene expression in aroid kinase A signaling, NGF, and KIT signaling pathways, indicating that the colonies from the same patient have differential gene expression signatures, with a significant subset expressing neuroendocrine-like signatures. Now, in addition to characterizing these heterogeneous CTC colonies, we have been performing some time-lapse imaging experiments as part of our pharma collaborations to better understand the relationships between CTCs and immune cells. And again, these were cultured under low oxygen and hypoxic environments. Essentially, what we did was incorporate a functional screening assay tied closely to genomic sequencing. Now, as you can see from this image, you have in one frame, which is about half a millimeter wide, a collection of dendritic cells, macrophages, and T cells interacting with individual CTCs. One of the tumor cells have been microinjected with the red fluorescent dextran dye to track the cell over time. So bear with me, I'm going to try to play this movie here. Now, you can see that there's heterogeneous collection of cells here. They're essentially drawn together, possibly through paracrine signaling. You can then use computer vision software to quantitate these relationships by measuring the number of T cells, dendritic cells, macrophages that are bound and interacting with the tumor cells. This, again, is just one simple example of the experiments that you can perform by enriching patient CTCs, but with the added ability to study them when they're alive in culture. This opens up endless opportunities for downstream experiments. Shifting gears, I just want to spend a few minutes just to talk about some of the projects that we're working with, in particular with Chuck Ryan and Eric Small over at UCSF. Now, we internally refer to this project as Liquid Biopsy 2.0. So this is where we're going to be characterizing ctDNA in the plasma by performing targeted sequencing on a panel of actionable mutations, complemented with CTC characterization using whole genome sequencing and mRNA-seq. We'll also be monitoring these patients serially, correlating against clinical outcome. The proposed study is an ambitious one, where we will sequence not only the CTCs, but the patient's immune cells, as well as any metastatic biopsies that become available. The general idea is that through serial monitoring of patient blood samples, we can establish a baseline mutational or gene copy number profile for a given patient, essentially monitoring the changes of up to 30,000 analytes to predict patient outcomes. So with that, I'd like to conclude again by thanking the organizers for the kind invitation, and I'll be happy to answer any of your questions.
That's great stuff. Really, really cool stuff. You know, one of the things that I couldn't help but note is those cells were AR negative, the vast majority. And PSA wasn't so predominant either.
Yeah.
So these are pretty undifferentiated. You seem to be growing this, and neuroendocrine, I'm not quite sure how you defined it, but I mean, these are some different beasts you're growing out. I have a feeling they're pretty nasty ones too. Okay, all right, just making sure I noted it right.
So yeah, I have a question about that underlying technology, like the success rate of collecting CTCs or getting clusters to grow. And do you have any idea, some patients more than others? You mentioned small cell, non-small cell versus prostate. Can you characterize that and tell me what the success or take rate is?
So the metric by which we're successful in being able to culture out of just peripheral blood is really the processing time of blood draw towards and plating on our substrates and putting it into a bioreactor. And so we've been collecting these samples in a 3-hour window, and we're getting upwards of 50%. That being said, again, not every blood sample could be processed within a 3-hour window. And so we're working on different sort of workflows to accommodate for shipped samples. But again, using Ficol-based centrifugation, the assumption is you lose half your cells. And so from 10 to 20 mils of blood, we're getting roughly anywhere between 25 to 50%. And we're, again, correlating that with disease burden as well. But we've been just focused on late stage at the moment.
Next.
Great talk. I had a question. You mentioned PD-L1 expression going up with the CTCs compared to cell lines. Have you looked at, in comparison to sort of the tissues, the primary tissue sample, or within the same patient even, different samples?
We haven't looked at biopsies, but that's something that we're obviously interested in. We have looked at it for other disease types, namely for non-small cell lung and pancreatic cancer. PD-L1 is very interesting. Different cell lines express PD-L1 differently, and that's again determined by the sort of the culturing conditions as well.
Right. So I was kind of curious whether it was related to the actual ex vivo conditions, or whether it's something that you are predicting might be happening in vivo in the patient.
That's a good question. And what we've seen is that across, using the same culturing conditions for all across patient samples, we're seeing variability. And so we think that maybe this might be a diagnostic window as to potentially making a candidate, or a patient being a candidate for immunotherapy.
Thanks.
So you brought up a series of very important questions. One was about the fact that we grow normal cells in culture, we use 20% oxygen, they're not on the pressure. But we also always stabilize the pH. We use CO2. And so the other part of the microenvironment is it's a acidic microenvironment. So in your systems, have you actually, it's not to make terribly acidic, but I mean 6.8 versus 7.2 does a lot of things differently to cells. Have you actually looked at pH change?
We haven't. That's one of the projects that we're actually pursuing in parallel with other factors. So the substrate that we're using, by no means is it fully optimized.
Well, let me come back to that, because you use some very important words. Cultured. In other words, you had cells that were in circulation and you got them to attach, and you showed us that most of the cells were dying. So the word culture is a little confusing. What are you defining as cultured? That the cell lives 8 hours? I mean, are you able to expand them? Do they grow? Or are you just putting them down on your hydrogel and then filming them for a short period?
So we're defining culture as the ability of viable cells to adhere to our sort of collagen-based hydrogel and propagate at least one round of replication over the course of 7 days. And what we're finding is that, again, there's variability amongst patient samples. But interesting enough, we're using, I think, sort of the canonical features of a metastatic tumor cell, in the sense that they have to be viable, they have to be adherent at some point to initiate new sort of tumor sites. And what we're seeing from sort of just simple Ficol-based centrifugation is that these cells typically are not alone. They're with other cell types, either with other tumor cells, endothelial progenitor cells, or other immune cells.
Well, they're epithelial cells. They don't like living alone.
Yeah.
That's their phenotype.
Yeah.
So, but let me ask you this. So when you do this, have you ever been able to take a specimen and do it under the conditions you're describing, which is the low oxygen tension versus 20% oxygen, to see, is there much penalty, or does it in fact help having a lower oxygen tension to have these cells attach and at least proliferate once?
So my background has been in primary cell culture. So a lot of this sort of, the protocol and the workflow has been sort of derived from the stem cell culturing field. It's pretty well established that to maintain primary cells over long periods of time, you really do require hypoxic culturing conditions. This is important for senescence as well as the promotion of stem cell factors.
Right, except that you showed that these were circulating cells that are in the only part of the body that's not hypoxic, which is the blood.
Actually, this in circulation, it does range around 15% to as low as 5% in the capillaries.
Yes, for a short, but as a whole, I mean, that's not a hypox, and it may be that they're going to do much better when you put them in that environment. Because I agree with you that one of the oldest things that I have never really seen anybody really get up and discuss is that when the Eppendorf microelectrodes to be able to measure PO2, a very interesting thing happened, right? It was that the normal prostate was hypoxic.
Yep.
And you go, okay. And yet, for example, glucose uptake, you would think if they were hypoxic, you'd be able to image all kind of stuff with essentially glucose,
Yeah.
but you can't, okay? So we're left with either the prostate as an outlier in that it looks like it's hypoxic versus it is hypoxic. And the figures you showed, I believe, and there's been really nice studies in the operating room where people have put this in and measured it, but it is remarkable then that all the cells that we ever, culture and that we're maintaining as our models are all in 20% oxygen. So I think what you're selling, not selling, what you're telling us, what you're telling us appropriately is that if we put them in that environment, we're going to get a readout, which will be essentially the readout of cells growing in a, essentially, not only is it not hypoxic, it's superoxic, essentially. And then yet, what we're talking about in the real people is something much lower.
Yeah.
So it makes you really wonder. So it would be really important, I think, to take your cells and see what happens in your essentially your arraying when you grow them in the way you're doing it with some of them, and then put them in 20% to see how much of a big difference that makes.
I think future studies just looking at taking these cells, putting them into animals or creating cell lines is something that a lot of our sort of partners have been asking for.
I am praying that you're right. I've been doing that since 1977. And I have over 500 different attempts, and I've gotten 5 lines in my lifetime. So, and what I have found is that the ones that would have established if I was a first-year graduate student or a Nobel Prize winner, because they are just either aggressive and they'll establish or they won't. And I've never been able to trick one that wasn't really very aggressive to grow. And I can get them to grow shortly, and I can even get them 1 or 2 rounds of replication. Yeah. But to be tumorigenic, that's another question.
Yeah. We should talk. Thank you so much.
Yep.